Hi, friends. It's Mr. Rogers here, and I hope that you guys have had a wonderful day so far. And we're going to cover prokaryotic cells really quick. We're actually not going to use my cell PowerPoint. We're going to use a different PowerPoint because I want to make sure that I pinpoint a couple things that make prokaryotes different. Usually, we kind of brush over prokaryotes really, really fast, and we're going to do the same thing this year, but we're going to kind of hit a little bit more. I want to hit this little tiny thing that's going to help us later on. And so, in 10th grade, you talked about prokaryotes versus eukaryotes. Prokaryotes are bacteria. They do not have a nucleus. Um, as you can see, I'll put it on the screen. Sorry, I should be doing that. They're unicellular. That's going to make them bacteria. There's a couple different. These are different shapes of bacteria. Right now, we don't need to worry about this. Um, we have size. They're tiny, very, very tiny cells. You can kind of see the size difference here. The internal structure, there's no internal compartment, so there's no organelles. It's just an open area. So imagine kind of like um, I once interned at a school for a brief moment that had no classrooms. It was like in a giant library type room, and there was no walls. And so you can kind of see like there's no specified areas. You can't say that this section of the cell does this or this section does that or whatever. Everything is kind of open, and so there's no membrane bound organelles. There is only ribosomes that could classify themselves as organelles, but we usually don't because they're not so much a compartment. They're just a specialized protein that reads mRNA to make more proteins. They're not so much a set room. And the other part about the no internal compartments is there's no nucleus. That's the main one. There's no nucleus in prokaryotes. And then there is circular DNA in prokaryotes, which is different. Our DNA and eukaryotes are on chromosomes, and we call it naked DNA because they're not wrapped around proteins. So they do not basically they do not store their proteins in an organized or store their DNA in an organized fashion we do. We'll talk about that when we get to DNA and genetics and whatnot for eukaryotes, but we store it in this around histones. They do not do that. And so, kind of skipping around this slide a little bit, the main thing I want you guys to know is obviously no internal compartments, no membrane bound organelles, no circular DNA, I should say, not no, circular DNA and naked DNA. But there's some other things on this, why I chose this guy, this PowerPoint. So the other one is because I wanted to point a couple things out. So prokaryotes have a lot of variations, and because they have no organelles, they do everything in that one room. There's no walls, and so everything is kind of mixed together. And in that one room, there is always intriguing amount of evolution that happens that can develop into different things. And so, for example, we might have here a photosynthetic bacteria that does the process of photosynthesis in a giant open space. Plants have to do it in their chloroplast. And we'll get into this with the endosymbiosis theory, how this all connects. But this is just doing photosynthesis in this big open space. There's not a set spot to do it. Or this guy is doing cellular respiration. It's a aerobic bacterium. He's using oxygen to make energy. Well, he's doing it inside of his cytoplasm, which is interesting, not an organelle. Okay. Now, prokaryotes also have an interesting cell wall. We're not going to get too much into it, but one day when you take microbiology or more in-depth biology, you'll learn about gram-positive and gram-negative bacteria. That's basically how we stain them, how we go about staining them, and what they actually have. It just basically depends on what kind of cell wall they have, or cell membrane, I should say. Some of them, you can see here, this almost has a double layer. This one has one. So this guy gets stained by the gram solution. This guy does not. Gram-positive bacteria respond better to antibiotics and most drugs than gram-negative. Just so you kind of know. It's not really that important for us at this moment. This one I'm not too worried about. Again, remember, bacteria has a wide ranging of different diverse ways of living life, and that's because each cell can have their own unique reactions that happen inside of them. They don't have organelles. Everything kind of interacts together. Now, let's keep going. Here we go. This is what I wanted to get to. This will help us later. So bacteria cells have something called plasmids, and plasmids are an interesting idea. And what they basically are is you have the main bacteria DNA right here in a bacteria cell, and that codes for the bacteria, tells the bacteria how to live and stuff like that. But bacteria also have small supplemental DNA throughout their cell that they do a couple different things for. You can see they're everywhere here. Now, all these different plasmids, they are released and given off and also kept inside bacteria cells at all times. And what they use them as is almost like trading partners. And so I have this little circular piece of DNA and I can give it to another bacteria and they can give me mine. And what that does is it increases diversity because now I have some of their instructions on how to do something and they have some of my instructions. Also, you can see there's plasmids everywhere. We actually find plasmids across the whole earth everywhere on almost every substance. And so if a bacteria dies, its plasmid stays there, and another bacteria can actually come along and pick it up, and it can become part of their DNA. And so bacteria also have a genetic engineering trait inside of them where they can at any point take a plasmid and they can implement it into their actual their own DNA that they don't get rid of. And so these bacteria can trade, they can morph into different things, they can pick up traits from other bacteria. It's a unique recombination that doesn't happen in anything that we think of as a living thing in a macro sense. Like we don't go up to a dog and give the dog some DNA and the dog gives us DNA and we become more like a dog. But this happens in the bacterial world every single day. So plasmids are very interesting. It's a huge part of bacterial genetics, and we're going to talk a little bit about that later on, but it's a huge bigger part of biotechnology and how one, we are learning how to basically use bacteria for good, and two, how we're trying to stop bacteria that are a problem for us from getting bigger and better. And it's an interesting process. And so I wanted to cover these things. I hope that helps. I'm sorry I kind of skipped around the PowerPoint, but I just thought it did a better job of explaining the things I wanted to explain than my other one. If you have any questions, let me know, and make sure you guys are working on your organelle chart, and you have an organelle quiz on Friday. And also today you guys got the cell group questions. If you're a virtual student, because you're probably listening to this, you're virtual. You're doing that by yourself, even though it's as a partner. So you get to pick your own, though. The other members of class, they get assigned them, even though they have a partner. So that's the difference between the two. And good luck with it. If you need help, let me know. Thanks.